Namaste to all. Welcome to the next session on drug discovery and drug designing. So in our previous session, we have seen about drug target identification and validation. So in that, we have seen about the mapping disease loci. So we have seen the basics, we have seen about the DNA, then how the DNA is being joining to form the nucleoside to nucleotide and nucleotide to how the sequence is being formed. And then regarding the sequence, we have also said that this is again forming into a compressed form, that is your chromosome. And in chromosome, if it is there, it can be in the different loci or it can be in the same loci, that is the linked part. So based on that, the important aspect in this is related to the gene tracking. So gene tracking means what? Basically that we have to understand. So in this complete session, we will be discussing more about the gene tracking and how it is useful based on our studies, how it is useful for the diagnostic aspects or application aspects that we will see in this particular session now. So gene tracking, if you see, this is a method for determining the inheritance of a particular gene in a family. So what happens is, if for example, if father or mother, if she or he is got some defective gene, then to identify that in the olden days, it is very difficult. So in nowadays, if a pregnant lady is there, okay, and after getting confirmed, that is on the 5th month itself, they can decide before the baby gets completely developed. Before that itself, they can decide in the 4th month or in the 5th month that if they want to go for the amniocentesis studies, that is the genetic studies of the particular baby, whether they have the particular disease or not, the karyotyping can be done and they can identify that whether the particular disease in their family it is running or it is being inherited to the next generation or not. This is the basic concept regarding the gene tracking. So it is used in the diagnosis of the genetic diseases, that is the cystic fibrosis and Huntington disease, and the restriction fragment length polymorphism, that is the RFLPs situated in or near the locus of the interest are identified using the gene probes and suitable markers related to the RFLPs selected. So what happens is, once you apply this restriction fragment length polymorphism, this particular study, you can identify that where it is present and near to that what are the different sequences are present. So all these things you can identify for the particular sample, and these can be traced through the members of the family and used to detect the presence or absence of the disease locus. Prenatally, there is a prenatally means before the baby gets born, before the baby gets developed. So the prenatal condition we used to say in future at risk pregnancy cases. So to avoid the risk pregnancy, so in some what happens is that there can be that baby development is taking place, but cystic fibrosis is there, or Huntington disease is there, or the you can say like Down syndrome or any other particular problems are there. Nowadays the solutions are there that they can go for the karyotyping or the genetic studies, and they can identify that how the particular baby is developing and whether any inherited disease or gene effects are there, which can lead to the different kinds of disorders or syndromes. So defining the gene tracking, you can say following a gene through a family by observing the inheritance of a marker which is being inherited alongside the gene. That means if, for example, one particular disease gene is there, nearby that particular gene, one more gene will be there which is supporting them. So yesterday, that is in the previous session, we have seen that is, if a gene is there which is defective, near to that gene also act as a marker gene. So what happens is we can identify that particular gene based on their linkage. So the knowledge on the genes helped in exploration, if you see, that is how much information a genetic test can give depends on the state of knowledge about the genes which are involved. But in principle, the laboratory analysis, or you can say the diagnostic method, if they want to apply like gene sequencing, karyotyping, RFLP and other things, the diagnosis can be made into two essential different ways. So either you can go with a laboratory testing, like the simple laboratory testing like gene sequencing and then karyotyping and other things, or you can also go for the bioinformatic tools and then you can go for the analysis part. So this can be done. In this particular study and the next coming topics, we will be studying about the algorithms or applications or so many things. So in that, the most important part in this you have to understand is what is meant by consultant. So consultant means the individual, that is not the necessarily the affected ones. It can be a normal person also who presents for genetic counseling and through whom a family with an inherited disorders come into medical attention. So the consultant, you can say the consultant or a consultant like that. So usually if you go to the genetic counselors, basically these are actually called the consultants, or you can say like a counselors. So what happens is when you go to that particular person, they will ask your family history. So the family history, they will ask that whether your family had the diabetes, whether they had the genetic problems, or they had any kind of symptoms, something like that, whether they showed any kind of issues. So all these things, based on that, they will mark it in their gene analysis, and then they will put the mapping of that, and they will understand that whether this particular couple who are going to have their baby will have a deformed baby or whether they will have a healthy baby. So based on that, the neonatal analysis, or you can say the prenatal analysis, is very much important nowadays, because the current scenario is such a way that because of the toxic chemicals, because of the pollutants, because of the adulterants, so many changes are occurring and these deformed genes are getting expressed. So that is the important point here by which the mapping disease loci has come into play. So the testing the genetic basis, if you see the testing the genetic basis, it depends on the two aspects. The first is called as the direct testing, and the second is called as the indirect testings or the gene tracking. Direct testing means a sample from the consultant is tested to see whether that is the RNA or protein or DNA, anything it can be. So it is tested to see whether or not he has certain genotype. That is, whether any mutation has happened, whether any deformity is there in that particular gene, or whether any gap is there or mismatch is there, all these things they will analyze, and typically a pathogenic mutation in a certain gene. So the test is of an individual and gives the information about the individual only, not from any other source. So based on that particular subject or individual, they will analyze the particular sample, and after analyzing the particular sample, they will say that, okay, this at this particular position some mutation is there, the chances are there that it can have the particular syndrome or scenarios in the future. So gene tracking, if you see, that is the indirect tracking method or indirect testing method. In this, what they do is the link chromosomes from a heterozygous parent. That is, see, the linked markers are used here. So the families, when they come to the counselors, what they do is, based on their studies, they will take the samples from the male and the female, and they will analyze. That is, the linked markers, they will analyze that. They will keep one marker in both as common, and based on that, they will see that whether they have got any inherited high-risk chromosomes from the heterogeneous parents or not. So based on that, they will analyze that and they will give a mapping of that. So the test is of a family. So here both the parents, that is mother and the father, both are being tested here, and gives the information about the segregation of the chromosomal segment in the family. So how it has been segregated. That is, they can go a back cross also. They can do a back cross. Back cross means from the mother, they can go to their father and mother's genes, and then they can analyze all these things, and they can from this side also, paternal side and maternal side, both the things they can analyze, and then they can give a conclusion at the end that when they combine their gametes, what will happen is the new baby which is being formed, how much percentage of chances are there? That is 1:4 chances are there, or 1:8 chances are there, or it is very similar. So all these things analysis, they do it and they give the report. So the basic idea in this is, we have to understand that these studies, when it is happening, that whether it is going to be it is going to give you the indirect testing result, or it is going to give you the testing result. So nowadays what they are doing is they first go to the indirect testing. So first they go to the consultant. After finishing the consultant process, they will go to the direct testing method. So from that, the samples will be retrieved. They will analyze the samples, everything. And from the direct testing, again, they come back to the same counselor for tracking it. So that will give you more data, and that will give you the more justification for the particular effects actually. So you can see here how the particular method is being followed here. So first what they do is they see the phenotype. That is, the phenotype means how the parents and both the parents are looking, or how the baby is looking. And then the parent recognition, laboratory testings will be done. So a few laboratory testings will be done. The clinical diagnosis will be followed. So based on that, the basic counseling on the recurrence of the risk, everything will be understood. And the basic of the pre-symptomatic or the prenatal diagnosis will be carried out. So here in this, the first thing is Mendelian part. So if you see Mendelian part, there are so many syndromes are there, so many testings have to be done for confirmation. So empiric risks, like yesterday we have said that third law of Mendelian laws, how it follows. So based on that, also it can be said that it is in the different loci or it is in the same loci. So we have to consider that. And the gene is being then mapped, then cloned, and again, most of the mutation, whether they have any mutations or not, they will be studied. So what are the things, what we get from this, if you see here, is maybe the possible by imaging, chromosomal analysis, biochemical tests, and also that goes back to the indirect test by gene tracking, then mutation scanning, then we are having the specific mutation scanning then. So these are the different things which is being followed here. And if you see the history of gene tracking, it goes back to the Mendelian, because Mendel was the person who actually did the gene tracking in Pisum sativum in detail. Actually, he worked a lot and he made it certain rules and regulations which still follows, and it is being implemented for the human studies also. So the first type of DNA diagnostic method to be widely used, and the most Mendelian diseases went through a phase of gene tracking and then moved on to the direct testing. So once the genes are cloned, so after the genes are cloned, they are again going back to the same state of direct testing. So the gene tracking using the linked markers still has its place in modern molecular diagnosis. So that is a very important topic, because you know that linked markers means the gene, affected gene is there, nearby there are certain genes are there which are always close together. So that acts as a linked markers, and based on the linked markers we can track that particular gene in the loci, that whether it is deformed or it is a normal one. So the gene tracking shrinks and the direct testing grows. So how it is? So within each year that passes, the role of the gene tracking is actually going down. Why? Because the consultant, by seeing the parents or by seeing the effects, they cannot say that this person is going to have it. So for justification, what they have to do is they have to go to the direct testing, that is the DNA sequencing, karyotyping, and so many other methods, neonatal screening is and other things. Then only they can say that, okay, this particular disease can happen here. It is not from the parent part, that is only seeing the phenotype of the parent. They cannot say that this person is going to have it. So that is why the gene tracking is shrinking and the application of the direct testing is growing everywhere. So however, the direct testing is always possible, and even when it is scientifically possible, it may not always be practical in context of a routine diagnostic services. So it is very difficult. Why? Because it is costly affair and it will take more time for analysis, because each time whenever they have to take the amniocentesis, that is when the baby is developing. From that they can take little amount of the skin, or they can take the little amount of the fluid where the baby is developing. So it requires the use of ultrasound, sonography, and experts has to be there, like gynecologist has to be there, and there has to be a government record that they have undergone this particular test or not. And for each thing, each sonographical test, they have to have the records. It is not that any doctor can do this. There are trained doctors are there. They can take the samples, and it is like a laparoscopic method. So they have to take a little amount of the biopsy like things. So from the amniotic sac, from that they have to take it, or from the baby which is developing, from that they have to take it. So it is a very risky thing. So that is why the experts has to be there. So many things are there while doing this, while doing or for giving for the particular sample. So the essential logic of the gene tracking, if you see, that is the distinguish the two chromosomes in the relevant parents, that is find a closely linked marker for which there are heterozygous. That means a closely linked marker you have to find, that is from the parents you have to find that which is very nearby, and based on that aspect only you can say that okay, both the things it is nearby means this is closely linked marker, and that can be used for both the parents and then for the baby. Then determine the phase. Phase means that is work out with the chromosome carries the disease allele, and work out which chromosome the consultant receives. So that is important. And the phase of the linked markers, if you see the relation, that is a coupling or repulsion between alleles at two different or two linked loci, it can happen. That means if allele 1 A1 is on the same physical chromosome as of the B1, that is if they are both are nearby, they are coupling. They are nearby means they are coupling. If they are on the different parental homologs, different parental homologs means, for example, this chromosome is from the mother, this chromosome is from the father, so one is in this and one is in this, means this is a repulsion. So they are always in the repulsion part. So this marker, so always we have to select in such a manner that it has to be a coupled marker, or you can say linked marker, or a coupling has to be there, that is in the same chromosome it has to be present. So steps in identification, if you see, so consider an example, that is a lady who is pregnant and wishes a pre-symptomatic test to show whether she has inherited the diseased allele in their baby, developing baby. So what they have to do is, the first step is to tell the parents two chromosomes apart. So we have to identify that in which chromosome the disease is, whether it is in the parent's chromosome, whether it is in the mother's, there is a mother or father's chromosomes. A marker closely linked to the disease locus is found for which her mother is heterozygous. So usually the scenario is it travels from mother to the child because it is developing. So maximum cases the mother is like a carrier and again gives back to the offspring. So here they have to check for the mother as well as for the father, and mother is screened mostly in a detailed manner so that they can identify that whether she is a carrier or not, or whether she is possessing something which is giving it to the baby or not. And the next established in this is the phase, that is work out which marker allele in the mother is segregating with the disease allele. So we have to note that it is a segregation pattern in the family and not the actual marker genotype. That is important. So both the parents has to be analyzed for this disease allele, and then based on that marking aspect, the child has to be the prenatal screening has to be done, and they have to identify that. So the prerequisites for the gene tracking, if you see the prerequisites in this particular cases, if you see, that is the disease should be adequately mapped. So they have to identify thoroughly that whether the disease is there or not, so that the markers can be used that are known to be tightly linked to the disease loci. So what happens is, if the disease loci is there, nearby the genes are there, so they have to identify or consider them as the marker, and they have to be mapped. So they have to be analyzed and they have to be screened properly. So what happens based on this? They have to undergo, once they are screening the parents, that is the mother and father, and the sample, that is from the baby, if they have taken any sample and they have analyzed it, they have to create a pedigree. Pedigree means that is a mother's father and mother, whether they had this, or father's mother and father they had this, or grandfather they had this. They had to make a pedigree, and from that they had to devise, that is how many siblings are there for the mother side, how many siblings are there for the father side, whether the father's siblings are also having any disease or not. All the things they have to study, and from that they have to get to the conclusion that whether the baby is going to have that or not. So that is called as a pedigree structure, and sample availability must allow determination of the phase. So it actually gives us a good result, just saying that whether the particular baby is affected or not. So the emphasis, this emphasizes the need for both an appropriate pedigree structure and informative marker type. So that is the DNA must be available from the affected child also, so that you can have an analysis saying that whether the mother is silent or father is silent. From that, whether who is giving that particular disease, no one knows. It is based on the environment also. It can be based on the particular chemicals also. It can be based on the particular kind of food also, or stress factors on the mothers also. So anything it can which can trigger this particular disease. So to rule out many things, they have to take the samples and they have to analyze the particular samples here. So this is how the pedigree is being constructed and it is being followed. So consider, and then this is how the DNA tracking is also being done and they analyze it. So if you see the first one, okay, so the first one, it is having it is having like disease one, okay. So here the mother is having the disease, father is normal, or you can say the mother is normal, father is having the disease. So the chances in this is maximum the female is going to be the carrier here, okay. So since father is there, mother is this one, so father is affected, mother is not. So in this, what happens is the boy who is going to come is normal, but the female which is formed here, that is going to be the carrier here. So carrier, since it is a carrier, this is the second generation. So what happens since he is the carrier? So when she get married, okay, when she get married, the babies which are being formed here, in that 1:2, that is 1 in 2, any one can have this particular, the boys can have this particular kind of alleles, by which the defective alleles are going to give them the particular kind of disease like cystic fibrosis, or you can say like neurofibromatosis, or different kinds, whatever the different diseases are there, they can, any disease can be happening here. So this is also being analyzed with the help of the DNA study, and from that actually they've been analyzed. So from this you can understand this. This is from a male. You can find that this is having the gene. This is from a female. So what is happening from the female? Both the genes are there. That is a normal gene is also there and affected gene is also there. Then what happens is when the son is being taken up, they have found that the normal gene is present. Along with that, the second child is having the defected gene. So from this DNA analysis also, you can analyze that whether the particular sample is normal or whether it is a deformed one. So this gives us a mapping and a pedigree structure, and gives us a good idea that how the direct and the indirect testing is giving us a good result for gene tracking of the particular loci. So the recombination sets a fundamental limit on the accuracy of the gene tracking, that is the recombination fraction, and hence the error rate can be estimated from family studies by standard linkage analysis. Standard linkage analysis means the parent has to be tested, and after that they have to give the report to that whether the baby is going to get or not. So that is what is the standard linkage analysis is. With almost any disease, there should be good choice of markers showing less than 1% of recombination with the disease loci. So 1%, yesterday we have seen the theta equal to 0.01. So based on that, we can say that farther, that is it is lesser, it is having the disease loci. So if it is closer, the disease loci is not there, or you can say. So the recombination between the markers and the disease can never be ruled out, even for very tightly linked markers, but the error rate can be greatly reduced by using the two markers loci situated on the opposite sides of the disease locus. So this is about this, and calculating the risks in gene tracking. If you see, that is the factors in assessing the final risk, then probability of disease markers and the marker recombination, then uncertainty due to imperfect pedigree structure or limited information of the markers. So uncertainty means if you don't know the the pedigree structure, whether the father is infected, mother is infected, their whole generation, if you don't know, then also it is a very difficult thing to track the particular gene if it is affected or not. Then uncertainty as to if somebody in the pedigree is carrying a newly mutant disease allele. That is, newly mutant disease allele means because of the stress, because of the induction or inducing of any new kind of toxin in their family, whether the particular generation has got affected or mutated and giving us the disease, we don't know. So that is the risk, you can say, in the gene tracking. So you can find the genetic risk calculation by the Bayes theorem. So it is basically the posterior probability. So the P A by B, if you see, this is actually P B of A, that is P of A by P of B, you can say. So this is the likelihood and this is the prior probability. So what happens here? So based on the calculation by the Bayes theorem, by the statistical method, you can come to a conclusion that, okay, this can be the chance, or this could be the chance, or this might be the chance that whether the particular baby is going to get the particular disease or not. Then the linkage analysis program. There are certain softwares are there by which the linkage analysis program can be done. So you can see this particular baby in which the limbs are being not formed properly. There is a cleft-net, there is a what you call the lips actually got joined with their nose, okay, and their ears are also actually deformed. You cannot say that deformed completely. One is closed, one is open, like that condition is there. So to identify this, they have to have the complete pedigree analysis of this. So if you see the complete pedigree analysis, in this you have to take the family 1 and family 2, and then based on that you have to do the analysis here. When we do the complete analysis, from that we can identify that whether the baby, how it is going to form. In whatever case it is, whether the baby is going to form like a deformed one or whether it is going to have the normal one by the karyotyping. This is all because of the karyotyping analysis, we can identify this. So coming to the summary of the gene tracking and the mapping of the disease loci, this is once a disease gene has been located using the linkage analysis, the DNA marker can be used to track the disease gene through the families to predict the genetic state of the individuals at risk. And prior to identifying the specific gene mutations, this can provide the information about the carrier risk and enabled the prenatal diagnosis in certain situations. So as we have shown in the previous slide, so we can prevent this kind of risk if we know complete history of that particular person, family and all, that mother's family or father's family, then we can identify that this disease can happen. If it is not there, then you have to go for the karyotyping or amniocentesis, or you can say prenatal diagnosis. And in some cases, what we can do is some child will be born. So what they can do is they will be looking healthy after the birth. So after about a month or after about a week of the birth, what they do is they actually take the blood samples, and from that also they can analyze that whether this genetic issues are there in their family or not, whether the baby is got affected or not. They can analyze after the delivery also. But best thing is before the delivery, if they know that in their family this problem is there, they have to go for the prenatal diagnosis. That is the best method. Then before the gene tracking can be used to provide a predictive test, family members known to be affected or unaffected must be tested to find an informative DNA marker within the family and to identify which allele is segregating with the disease gene in the particular kindred. So here what we have to understand is once they identify this particular marker, that is the affected marker or a gene marker, so what they can do is they can use this in their family for the next generations and coming up generations, because that will help them to analyze that whether the particular family is got affected or not. And because recombination occurs between the homologous chromosomes at meiosis, a DNA marker that is not very close to a gene on a particular chromosome will sometimes be inherited independently of the gene. So sometimes it is also there that when they are very close by, they can go to the next particular scenario. So you know that when the meiosis takes place, there are 4 gametes will be formed. So in this particular case also, in one of the gametes, it can happen like they have independently inherited and it has caused the problems. So that we have to analyze here. The closer the marker is to a gene, the less likely is that the recombination will occur. In practice, markers that have shown less than 5% recombination with the disease gene have been useful in detecting carriers and prenatal diagnosis. And there is always a margin of error with this type of test, and results are quoted as a probability of carrying the gene and not as a definite risk. That is the important part, because statistics is mostly used here. So statistics, as per the theorem, they say that 80-20 ratios. So 80% chances are there, 20% you cannot say that this is going to be the result. Okay, so always there is a 80-20 rule, or you can say 90-10 rule, so which follows always in the statistics. So this also applies for this karyotyping experiments or prenatal diagnosis. Then linkage studies using the intragenic markers provide much more accurate prediction of a genetic state. But this approach is not only used now when the mutation analysis is not possible, as in some cases of Duchenne muscular dystrophy, Marfan syndrome, or neurofibromatous type 1. So these are the different diseases which can, or syndromes which can occur if it is not being diagnosed properly. Because if the linkage markers are not being created, then what will happen is they will leave like that. When the baby is born, no one can blame that particular gynec doctors that this has happened because of you only, because they have to undergo all this testing. That is why the doctors have now made it very mandatory that they have to undergo these all testings, that these all scannings, these all testings and all, and then only they can go for the particular delivery. If they find it in the 5th month that there's some problems are there, then they will go for the genetic analysis or prenatal diagnosis, and then they can have it. So with this, I hope that you have got an idea that how the gene tracking or the mapping of the disease loci is being done. Hope so that it will give you an insight for the next session. So thank you and have a good day.